All right. Seems like people are still hopping on, but why don't we go ahead and get started? Thanks for joining everyone today. Um, we have Stephen Brenner here from UC Berkeley today. He's going to talk to us about CAGI, which is critical assessment of genome interpretation. I don't think you've ever presented on this forum before, which seems surprising considering how relevant this kind of work is to the folks that often join this call. So we're really excited to hear about what you're going to present today on the CAGI initiative.
Great. Well, thank you very much for the invitation to join here. I think the fact that I haven't been here before reflects that we haven't been, as well informed about cancer as we would like to be, and this, I think, really a good opportunity for us to try to connect these communities together. So CAGI stands for the Critical Assessment of Genome Interpretation, and we're motivated by the fact that there are hundreds now of methods for trying to predict the impact of a variant. We have a database called VICC, which categorizes about over 400 of them right now. We're about to make a new release, which will have many more as well. The basic idea, which I think all of you are familiar with, that we want to often go from looking at a variant which we've seen either germline or somatically, having some sort of understanding of what that variant might be doing, and ultimately be able to predict whether that will be pathogenic or benign. And there have been these hundreds of methods developed for this purpose. But the large number of methods, in some sense, has made it more challenging to figure out how we should use them and when we should use them. And so starting about 15 years ago, we launched an experiment called the CAGI experiment for critical assessment of genome interpretation, which was modeled on the CASP experiment, which for protein structure, in which experimentalists would solve a protein structure, but before that protein structure was publicly available, the sequence would be shared with computational biologists who would then make predictions of that protein structure. And then once the structure became available, that the predictions would be compared with the experimental structure to determine where there had been progress in the field, what methods tended to work well, and which ones did not. And so we developed a experiment which was analogous to that for trying to understand how we could predict the impact of genetic variation. This is a slide where the first 5 bullets were actually identical to the slide as I made it back in 2010. I did add the 6th bullet more recently. Our goals were to understand the state of the art, to understand how good these methods are, to be able to tell when there's been progress and innovation, to be able to reveal where there were limitations with the methods, and help guide future effort to the areas that were most important, to highlight new challenges that were practically important, and to collaboratively develop new approaches. And on highlight new challenges, I want to actually maybe talk a little more about that, that there's been a continual kind of interest in the computer science field in applying methods to biology, but often the computer scientists don't know quite exactly how to move into the field and will often kind of develop toy problems which don't really solve real biological problems. And part of our goal here was to have a well-posed question that a computer science group would be able to grab onto to be able to understand how to actually enter the field and make productive contributions. We've seen many groups actually enter the field through this mechanism. And then finally, it turns out that today it's in many cases easier to develop a semi-decent method for predicting impact than it is to actually assess how well it does. And so a major part of what we've done is actually work on how to advance the science of doing assessment. The basic approach for CAGI is that we have several phases. The first one is we try to collect data sets where there's some sort of relationship between genotype and phenotype that has not yet been made publicly available, but is definitive in some way. And the phenotype could be something that is like a biochemical phenotype, or it could be something which is clinical, a patient's experience. And so this is a little different from what had to be done for CASP, where for CASP, simply they could look into the PDB, see what structures have been deposited but weren't public yet, and basically go and identify those. All of them involved a sequence and a structure, so it was a relatively simple data. For CAGI, we have to actually go out and figure out who has data that's not published yet and find those individuals who have those data sets and work on developing those into challenges. And the challenges often have very different structures. And engaging with Civic and with the VICC, we hope to be able to maybe be able to advance how we do this data set collection for the CAGI experiment. The second phase is the prediction season, where the data sets that are not yet public but the variants within them are public, are shared with the CAGI predictors. I should rephrase that. Actually, the variants themselves may not be public either. That the data predictors always sign a data use agreement that limits their ability to share the data until it's public, to be able to keep all unpublished work completely private, if that's what the dataset providers want. Typically, we run the CAGI experiment every other year, and we tend to have a prediction season that runs over the summer. So we're actually just wrapping up the prediction season for CAGI-7 now, although there are a few stragglers that are going to be running into the fall and even running into early next year. And then that's followed by an assessment season, where the assessors look at the truth data set of clinical or experimental data and compare that to what the predictors had done. So our assessors are busy at work as we speak. We use independent assessors. That CAGI leadership does not do most of the assessment. It's actually other members of the community who tend to do this. And then finally, every year, or roughly every year, we run a conference that in a year where we run a CAGI experiment, that basically that conference will basically report on what the findings of the CAGI challenges were. In interim years, we have what we call a CAGI Star Conference, which talks about kind of bigger scale issues and sort of what direction to go in the future. And so we'll be running a CAGI conference this year at the campus of Northeastern University in Boston from the 6th to 8th of December, where the assessment results will be presented along with contextual information from patient advocates, from clinicians, ethicists, and the dataset providers. So we then publish our work. We've had so far 82 publications in 3 special issues, as well as a integrated paper, which had 457 authors on it. Today, the CAGI consortium is now up to about 1,000 authors. I'll tell you a little bit about kind of what the data looks like. But one thing I want to emphasize is this is something we adopted from the CASP experiment, is that CAGI is an experiment, not a competition. That in general, we don't have any single unique top performer for a challenge. We're not looking for winners and losers, but rather we're trying to understand how the methods work and when one user would want to use what method for a different application. And it turns out that there are different goals that one might have in using one of these computational methods, and the goals in developing one, understanding kind of what we can discover from the biology, may be different from what one's trying to do when one's trying to research what genetic variants do, which again is different from diagnosis and screening. And so any given assessment might actually use a dozen different assessment approaches, and then we basically select interesting methods that performed in an interesting way to be highlighted, rather than looking solely for what has been the single top method on a given challenge. So we've been running the CAGI experiment since 2010. We've run a total of 7 challenges so far. The first one, which was CAGI-1 in 2010, had just 3 challenges. But from the very beginning, you can see here, we had some cancer genes involved that Sean Teftigian gave us a case control data set on CHEK2 to see whether people could predict how well basically predict risk alleles in CHEK2. And we've continued to include cancer challenges through CAGI-5. These are challenges from CAGI-6. I won't dwell on them. And I just got these numbers yesterday, which are the number predictors for the CAGI-7 challenges that have closed so far. The first 3 challenges shown here are what we call annotate all challenges, which are annotation accumulation assessment exercises. I'll talk a little bit more about those later and why those are special. And then these are challenges that have closed so far for the CAGI-7 experiment. Most of these are missense challenges. I'll talk a little bit more about how those work. And you'll see here that we actually have 3 of them that are associated with cancer, including one that we're very excited about, FGFR. And I'll talk a little more about that one also.
Can I ask,
yes.
how are the genes chosen for that previous slide?
Yeah, so these are basically, again, what we had for CAGI-7, that we basically go out and try to find what are the data sets that people have that will be unpublished but ready during the prediction season. So they need to be basically ripe, but not yet spoiled. And so we basically have a community of people who we know tend to have data sets, and that community now is about maybe 100, 150 people. And we reach out to them and see what would be good data sets. And so basically we start out reaching out to, again, the community is maybe 150 people of data set providers we have. We reach out to a large number of them and try to figure out what would be the most interesting things to do for that CAGI experiment. And so this is the data set that we came up with. We do try to always highlight cancer because we think it's a very important area for genetics. And we also think it's an area I'll talk about that's been underserved by the computational methods in many ways. So the most common types of challenges, actually, did I answer your question?
Yes, thank you.
Great. And if anyone else has questions, please jump in. That I've tried to make this not fill all the time, but make sure there's time for questions. And it'd be great to have them actually as I'm presenting rather than just all at the end. So don't hesitate to jump in. And I'm not seeing the chat. So if actually someone's able to look at the chat, let me know if anyone puts in the chat. That would be great. So a typical type of challenge we run is a missense challenge. These are challenges that are at a biochemical level, most commonly, where we're looking at a particular protein and its gene, and looking to see what the impact is of various missense mutations. So here's one we ran a couple of years ago, which was for mutations in NAGLU. This was a enzyme for which a company BioMarin had been making mutations. It was actually an interesting goal they had. They were trying to figure out actually what is the actual incidence of this disease to let them know whether it made sense for them to try to develop a drug for this disease. They knew how many kids actually get diagnosed with it, but their suspicion was that it was much more common, but the kids were basically dying before they would get properly diagnosed, and therefore that they thought the market was larger than the incidence would indicate. And so NAGLU causes this condition called Sanfilippo syndrome, which is a very neurological disease, and they made mutations or paid to have mutations made in a large number of variants that they were able to see as rare but present in databases like Nomad, and see what the impact of those would be on the activity of the enzyme. And so here's an example of one of the particular mutations that was made, GLU128ASP. So we basically provided that mutation identity to the predictors. The predictors made their predictions, and each of these dots here is a prediction from one group. So you see that most groups predicted that it would have about 100% activity. One group estimated the enzyme would have 50% activity. Some groups estimated intermediate activity. And then the black line here is showing you the actual experimental data, showing that in fact that this mutation had no impact on activity. The enzyme had about 100% normal activity. And so if you look at a large number of these, you get a picture that looks like this. So we also have many different ways of looking at the variation, but often it's helpful to actually look at exactly what people did. And you can find almost every pot combination of different types of results here. So if you have here's another mutation where the enzyme had about 100% activity. And again, most groups predicted close to 100% activity, but some predicted lower levels of activity. So this is a pretty good set of predictions for most groups. Here's one where the enzyme was dead, and most groups did correctly predict that. Here's one where the enzyme had sort of intermediate activity, and people's predictions were all over the place. And down here on the left, this P216R is one where most groups thought the enzyme would be totally fine. In fact, the enzyme had about 50% activity. So this is a result from a few years ago. And what we can see here, and this is typical of what happens with the missense challenges, that newer methods work better than the older methods. So the blue methods are newer methods. The brown methods are older methods that are actually about 20, 30 years old. That the results are almost always very highly significant. But then when you dig into it in detail, if you think about how we might use this clinically, things don't look quite as appealing. And so this is for the very best method on this challenge, the predicted activity on the bottom, the experimental activity on the left. And while there is a very strong signal, as I showed you before, that was highly significant, that your desire to diagnose someone or treat someone based on this genetics would be very limited. And this is a really big concern, as right now there's a lot of efforts going into newborn screening via sequencing, And the idea is you would identify newborns potentially with the disease by looking at the genetic variants that they hold, and then you would give them a treatment. And the idea of the treatment is you want to intervene before they show symptoms, and that treatment would then prevent the emergence of symptoms. But given this type of relationship, that one might pause before wanting to use computational methods to try to interpret those genetic variations. But if you look more carefully at this, actually, there's something that's very interesting. That actually over at the left-hand side, where the predicted activity was less than 0.1 or less than 10%, then in fact, basically, it was always right. This method pretty much 100% of the time was able to make the right prediction. And this is especially interesting because the disease actually only manifests itself when you have below around 10% activity. So in fact, it worked almost exactly where you'd want to. And now there are these enzymes here where the predictions were wrong. So there are definitely a lot of false negative predictions that were made. But where it did predict that the activity was going to be less than 10%, it was basically always right. And so you might, if you were saying whether you want to make a prophylactic diagnosis, this would be something that it seems like you might be able to trust. Based on these types of results, we built on this, and I helped create a subgroup of the ClinGen sequence variant interpretation group that was focused on computational methods. And this led to a recalibration of computational methods, as well as a clinical recommendation for the use of these methods, where historically in clinical use, these only could be used up to supporting level evidence, which is extremely minimal. And that now they can be, like orders of magnitude stronger, going up to potentially strong evidence even. And we feel very confident in the calibration that's been done here. And in fact, we think these are some of the most reliable data that one might use now in clinical variant classification because these have been so carefully calibrated. And I'll just acknowledge here that the members of that group so this was an effort that was distinct from CAGI, but it was sort of motivated by these results from CAGI. And CAGI actually helped support both the meetings where these met and some of the travel for these individuals for us to get together, which was critical for activity. The CAGI experiment also helped us understand limitations of the methods. One of the things that's interesting you can see on the right-hand side figure is that invariably the computational methods are more correlated with each other than they are with the enzymatic activity. And this is true even for methods which are extremely different in the approaches they use. And so on the left-hand side, I'm showing two different methods that were two of the best methods in the CAGI experiment that year, but they use really entirely different philosophies for how they work. And what I'm plotting on each axis is the prediction from the method minus the experimental value, so basically how far off they are. And what you can see is they're both off in very, very correlated ways, despite using very, very similar, using very, very distinct approaches. And we actually wonder in many cases whether this may be that whether the enzymatic activity that's being reported in this assay may have biases. There have been several cases where we found that actually the predictors were correct at predicting pathogenicity, but that they would miss predicting the enzymatic activity because the enzymatic activity would actually be poorly correlated with the pathogenicity. So in some cases, computational methods may actually exceed the experiment in modeling the actual pathogenicity and clinical impact. So as I mentioned in the CAGI experiment, we have been interested in looking at cancer variants. Most commonly, we've done this by looking at the cancer variants in genes like BRCA1 and BRCA2, and basically looking at loss of function in much the same way that we have looked at the variants in other genes. This is one of the challenges we had a couple of years ago, where the NICA consortium was about to release a large set of variants that it had characterized, and we were able to basically get those shared with our data, with our predictors to get predictions in before those made public and see how well computational methods could work on those. Amanda Spertle was really a key person that enabled us to be able to do that with the Enigma consortium. But we've also tried to look at more distinctive features of cancer as well. Back actually in 2011, we ran a challenge with Joe Gray where he had basically a number of breast cancer cell lines and treated them with a number of different agents. And it was, I guess, 54 different breast cancer cell lines, and a large number of different treatments. And the predictors were asked to try to predict which um, which cancer breast cell lines would respond well to which drugs, based on the genetic variants they had and based on the nature of the drugs. And so we were interested in considering doing more challenges which have these unique aspects of cancer as well. And I'll pause here. I don't have a slide about it because it's brand new, but I was really, really excited this year that we got this FGFR kinase domain challenge. This is a data set from Sven Diedericks, who did saturated mutagenesis of the FGFR 1 through 4 kinase domains. It's a paper that actually is in press right now in Science. And we were just managed to squeeze in that challenge during the prediction window. And what makes me really excited about it is because this is a gain of function challenge primarily. That's the core challenge, is to identify which variations lead to a gain of function that they were able to measure, because there are very, very few computational methods that actually look explicitly at that. As I mentioned, there are over 400 variant impact predictors. Of those, only 2 or 3 really seem to be focused on gain of function. A few of them, or more of them, will incorporate information about gain of function, but not very explicitly. The other aspect of that FGFR kinase challenge that makes me very excited is they also had pharmacogenomic information where they knew what was the drug response of the various variants. It wasn't looking at as many drugs, but just simply seeing just was it responsive to each of 2 different drugs. And this, again, is, I think, something that for which there's a great need, and there's very, very few methods that have been developed for this. That we ended up getting, as I think I passed by quickly, in the end, 9 different groups predicting for this challenge, which is not a great showing. We did not think we would get many. In fact, I thought we'd get fewer than that. In many ways, our main goal with this challenge was to demonstrate that there's a real need in this area and to motivate developments in this field to be able to build more challenges. Should I see there's something in the chat? Let's see.
I think it's
okay. Yeah, so we actually...
I have a question about this.
Yes.
So it describes below this business with the response to a panel of drugs, but that's distinct from the gain of function aspect of the experiment.
So this challenge that I'm showing you right now, that was one from 2011 that was done with Joe Gray. So this one was just where we had cell lines and he had the mutation and he had the transcriptomes, and the only challenge here was to predict the response to a large number of different drugs, on basically to what extent the cells would die, basically. That for the FGFR challenge we have this year, and again, I'm sorry I didn't make a slide specifically of this, that basically he has done saturation mutagenesis of the kinase domains of these 4 proteins, and he's identified which variants are gain of function, which variants are loss of function, as well as which of these are responsive to 2 different medications.
Okay, and so the gain of function there comes from the saturation mutagenesis experiment where they measure like cell proliferation or something.
Exactly.
Yeah, that's really cool. And sorry, which group was that data from?
So this data came from Sven Diedericks, who is in Germany, Freiburg, I believe.
Cool. Yeah, that's really cool.
Yeah, so we're really excited about it. Again, that I thought we would not get any predictors, or only get 1 or 2 predictors. I was delighted we did get 9 of them, but this is something that there should be dozens of groups that are caring about. And one of the key things that we're doing with this challenge is trying to emphasize that this is an underserved area computationally. We picked as the assessor for this challenge Russ Altman, in part because we thought that he'd be able to well articulate the importance of both this problem as well as his own research. And he's interested in pharmacogenomics more from a germline aspect, but we thought he'd be able to articulate well also the relevance of it in cancer as well. And so really what we're trying to do with this challenge is emphasize the field, emphasize the need here, and try to draw people into working on this for challenges that we run in the future.
And this one that you're mentioning is part of CAGI-7, the FGFR?
The FGFR is part of CAGI-7. So that challenge has now closed and is being assessed as we speak.
And in order for like a predictor to be part of the experiment, does it need to be public or private, or and is like the identity of those things revealed in advance, or do you learn about it all after the fact? Or because I
So
guess I'm wondering what are the nine, and like could we learn more about them now, or do we have to wait?
so I can't share anything until we basically make this estimate public at the CAGI meeting that will be in December. That so when people participate in CAGI, they have to sign a data use agreement, and basically that's in general the only constraint. Some data sets are more sensitive. I'll show you some clinical ones coming up which are more sensitive. But for this data, basically, if people sign the data use agreement saying basically that they're not going to misuse these data, they're not going to leak these data, that anyone can join. That in general, groups are named and identified. In rare cases, people can choose to be anonymous. If they are anonymous, that then they will basically remain anonymous, even if they do well, except for if they're a very top predictor, that then we have the right to reveal who they are, even if they want to be anonymous, if they're one of the very best methods. The reason we allow anonymity is that sometimes there'll be a group that, has some really wild new idea and they don't really expect to do very well, but they're curious about its performance and they don't want to sort of take on the reputational risk of having their wild new idea tarnish their reputation. But in general, most people end up being identified. Virtually everyone ends up being identified in the CAGE experiment. So you will learn who these 9 people are. They will probably be named at the CAGE meeting in December, and certainly in the publication that will follow, that unless any of them had chosen to be anonymous and end up not doing well, that information will be released. I'll mention that in addition to those 9 that submitted, we also have the assessors recruit other methods that are in the field, and they can run them themselves. So without naming names, there are a couple of there are a small number of methods that are known in the field for looking specifically at gain of function and for looking at variants that are involved in the cancer. If the method developers for those methods did not participate in the CAGE experiment, that then the assessor may choose to run those methods themselves and see how those methods perform relative to them. For example, I showed you on the previous slide how some new methods compared to very old ones, SIFT and polyphen. The developers of polyphen did not actually make submissions, rather the predictors ran those as well.
And are there any rules about the availability of the methods?
So we require the predictors to share with us what a summary of what their methods are, including information about how much was manual work, but we do not currently require those methods be publicly available. I'll mention something for the clinical genomes in a moment where we're doing something a little bit different, and we have thought about requiring the methods either be available or that they be automated. And actually for PRS also, that they need to be somewhat automated because of the way we do the evaluation of the polygenic risk score challenges. But in general, it's been whatever method you want to use, you're allowed to use it. And that's sort of following the model that was used for the CASP experiment, where sometimes people would do it entirely manually. And actually one time that did really well when Lexi Merzen ran a challenge that way.
It has to be
And just,
computational, though, what they're doing. I mean, could someone be doing something experimental?
um, in theory, someone could do that. And that could be done for CASP also. In fact, I actually tried to do a semi-experimental submission to CASP one year. There's nothing that prevents that, but you'd also need to declare that. You have to describe what your method is when you make your submission. That my hypothesis for CASP was actually that there'd be some very high throughput data we'd be able to collect from doing cross-linking mass spec, that you'd actually be able to do this in the time frame of the CASP experiment and see whether that can actually perform well. So if someone were able to do experiments fast enough that that they were able to show that things did well at making these types of predictions, I would be interested to know about that, but they would have to declare it. To Andy's comment, we actually did literally spray paint a Berkeley building. We really felt when we were working on trying to do this ClinGen clinical recommendation, that one of the major challenges we had was convincing skeptical clinicians that the computational methods actually were validated and could be relied upon. And in many ways, we felt like barbarians at the gate. That now, I think that there's been, by at least the leadership of the ClinGen group, a widespread recognition of that this calibration has worked well and that doesn't lead to bad outcomes. But certainly there was a lot of distrust because historically the methods have been overhyped and that the default thresholds for these methods are far, far off. That what's published for how you should use them is very different from what we learned you actually should do. So that's part of what we learned from CAGI, and then we apply that into the ClinGen environment for that formal classification. Any more questions on this? I'll move on to a slightly different topic now. So another type of challenge that we had, and I'll just mention this briefly, are clinical genomes. We've been running these for many years. We ran them in KG4, KG5, KG6, and KG7, where we would give out to, and actually we even had some complete genomes we gave back all the way out in KG1 from the Personal Genomes Project, where for the clinical genomes, though, we give people our genomes of individuals who are known to have some serious disease, which is believed to be genetic, and see whether the computational biologists are able to identify what are the relevant variants. And when we give these challenges, we'd usually have some of the genomes where a clinical lab had identified what they believe the diagnostic variants are. And so this is a test on basically, can the computational groups be able to recapitulate what we believe is correct from the clinical laboratories? But also there would be unsolved cases as well, where the clinical lab had gotten the genome and hadn't been able to figure it out. And in every single CAGI experiment, there have been cases that were solved by the community that were missed by top clinical labs. That the Sick Kids Hospital in Toronto ended up having a couple of cases solved. This was actually a really interesting one where the lab had not been able to identify what was the variant responsible for the condition, which was a type of epilepsy. That a predictor identified these particular variants in this gene as being what they thought were responsible for it. And so how do we know whether actually the predictor's prediction was correct? We have to do follow-up. And so in the case of this individual, that variants in this gene are known to cause a very specific type of seizures. The lab did not know whether the individual had that type of seizures. They had called up the clinician and said, what type of seizures did the kid have? It turns out they had the exact type of seizures you would predict for variants in this gene. And that was basically therefore deemed a solved case that had been missed by the lab. In the last CAGI experiment, we had data from Heidi Ream and Madonna Loria, which was assessed by Sarah Stenton, with data from the Rare Genomes Project. Again, some solved cases, some unsolved cases. And again, there were a couple cases that had been not solved by the Broad as part of this Rare Genomes Project, where they really put a lot of attention in trying to figure them out computationally. Predictors figured them out. Then the group at the Broad did follow-up studies to actually assess if those look correct. And indeed, there were a couple new genetic diagnoses that were solved there. So the clinical genomes challenges are ones where we do have some more restrictions on the data sets. There needs to be an institutional signature now on those due to the sensitivity of these data. But again, this is otherwise open to everyone except for where there's export control restrictions. And we're running that challenge again this year. It basically getting the agreement for the sort of legal transfer took longer than we hoped. And so it's actually just opening now and we'll be running through February. One of the things that's actually really cool about what we're doing this year is in addition to having it open to the broader computational community, that Heidi Ream has worked with a large number of clinical laboratories to run these genomes through their standard clinical pipelines on the platforms that they license. And we'll be able to evaluate, how about a dozen different commercial clinical lab platforms work as well by essentially recruiting those platforms into the challenge. So this is sort of analogous to how an assessor can run some popular method like SIFT, polyphen, or CAD that may not be the best method, but it's commonly used. For these platforms, the assessors typically don't have access to them because they have to be licensed, but that Heidi Ream was able to basically work with the laboratories to get them engaged. And then I'll just briefly mention that CAGI has also been involved in developing some innovative ethical approaches for how to work with these types of data. Essentially, all of the data we work with is legally and technically not human subjects data. And so there's no IRB relevant, that there's no sort of ethical oversight that's formally in place. But we felt that because almost all the data we work with here is derived from human individuals who had agreed to be research participants, that we had substantial responsibilities for stewardship and responsible use of these data. So we created a innovative approach called the Ethics Forum as a group of ethicists, patient representatives, lawyers, researchers, to come together and discuss what is the appropriate use of these data. And this has helped us make sure that we don't do anything that especially would be inadvertently inappropriate. That they've also actually, in some cases, motivated us to do challenges which we might have hesitated to do, the argument being that the participants had shared their data because they wanted research to happen, and that if we didn't actually do that research, we'd be undermining their goals. And that the risks, while not zero, were small enough that the risk of undermining the goals of the individuals who share those data was greater than the risk of data, for example, leaking in some way. And then the final challenge I'm going to mention in a little bit of detail is what we call the A9 challenge, in part because this is in some ways what I think might be a model for how we might work with CIVIC and work with the cancer community more generally. So the A9 challenge, it stands for Annotate All Amino Acid Alterations, Annotation, Accumulation, Accuracy Assessment. It's one of its category of what these annotation, accumulation, accuracy assessment challenges, where what we do is we ask predictors to make predictions for every single possible missense variant in the genome, and there are about 82 million of these possible missense variants, and these basically get saved into a form like DB-NSFP, which is commonly used by clinical groups to interpret genetic variation. And so they make predictions for everything. And then they make a submission on a given date. And then we freeze those predictions that have been submitted. And then we look to see what variants get classified definitively in ClinVar or get reported in HDMD after that date. So variants that essentially get classified and we know what their role is, but were not known at the time that the predictions were made, and so they couldn't have been used in the training of those methods. And so this actually models exactly what one wants to do with these methods. It's, could it have predicted a variant that became of interest at some point after the method was developed? And we then assess how well the methods do. Again, we use many different types of assessments. This is a rock plot, which rock plots tend to have a lot of problems for doing these types of assessments, but it's one that's familiar to work with. And what you see here is that in the most recent one that we ran from CAGI 6, that alpha missense ended up being the best single predictor. We have a separate plot that has meta predictors with Revel and things like that. Revel performs about as well. But what's interesting is actually while alpha missense does nominally the best, that this method here, VEST4, which is a method that is now almost a decade old, is only a little bit worse than it. This method 3 billion, actually, this comes from a company that's in Korea, and their participation in the CAGI challenge here was actually one of the key things that's helped that company expand, because they're able to show their method is one of the leading methods that's in the field. And so we would really like to consider, doing something similar to this with CIVIC, where there are variants that have not yet been classified for cancer. People would make predictions of what the impacts of those might be, and then we would, as those variants do get classified into databases, we would then assess how well the methods work on those. But as we've discussed, I think several times, that cancer has many distinctive features that are more complex than just looking at loss of function variation, which is principally what's being looked at here. Not really looking at pathogenic versus not pathogenic, but primarily this is some sort of loss of function type activity or impairment of function. And that what happens in cancer is more complicated, and we wanted to talk about how we might work with that. And so I think I'm just going to wrap up there. This will take a long time to scroll by. It's a list of the predictors in the predictors and participants in the CAGI 5 and 6 experiments, the 457 authors, as well as the more recent participants. So thank you for your attention, and I'd really like to discuss how we might potentially work together in being able to do additional assessments of methods developed for cancer, and actually also promote development of such methods as well.
Thank you. I have already asked several questions, so I want to give anyone else a chance to comment or ask questions now.
Hi, thank you so much for your presentation. I'm wondering, what kind of outreach I mean, it looks like you have lots of collaborators. I just don't want to like duplicate any effort to get you connected. But what kind of efforts have you done to outreach to find collaborators for CAGI? And I'm just trying to think like how could we promote this for you?
So in terms of finding data providers, we rely on two things principally. One is our program officers. One of them is at NCI, and so they've made some introductions for us. In addition, that we have a advisory board, and that includes people like Mark Greenblatt and Sean Teftigian, and they've helped us be able to connect with people who have data sets in the cancer field as well. But I'd say that we haven't really, been as connected as we would like to. My own research is not in cancer, and so I both don't really know who's doing, the most exciting things or who has the most relevant data. And I think even more importantly, that I'm really kind of, only guessing at what I think are the most important challenges for computational methods in cancer. And so we would really love to have, more insight and more feedback. And then in terms of trying to engage predictors, I mean, we don't know of that many people who are developing methods specifically for cancer. Rachel Karchin is one of the people very actively involved in CAGI, and obviously someone who's been, I think, a pioneer in developing those methods, that she reaches out to people that she knows. We have various mailing lists, and we have Blue Sky and X accounts. But ways to be able to advertise this better would be something that we greatly appreciated.
Yeah, that's great. I know that a lot of people are using kind of methylation as like a diagnostic predictor or different kinds of signatures as predictors in cancer. That might be kind of a route to go too. But yeah, definitely. However we can help, I'd be happy to promote this.
Yeah, so I mean, there are many ways in which we would like help. One is helping us identify, what are actually the most important problems to be solved, that especially those that are distinct to cancer and different from, one might do for other diagnostic genetics. Second is, help leading us to people who have data sets that are not yet published, trying basically always become, one step ahead of knowing who has data that's not yet public. And I just don't know the field as well in terms of those individuals. And then third, how we might be able to engage people to predict.
Yeah, thanks for the presentation. I was thinking about it. I think some of it is obviously challenging just due to like the nature of the data not being needing to be public yet. I've had many chats with Malachi and Obi about predictors, especially with one of the ClinGen SEV-CEPs that we're on, and really trying to leverage like in silico predictors for like gain of function mutations, for instance, and how that's, like you mentioned, really not what they're trained on or designed for initially. And so at Nationwide Children's, we even actually submitted a list of genes to the AVE Alliance to try to get them to start working on a couple genes that had more like gain of function missense mutations that we routinely kind of struggle with a little bit more in terms of just like what is the actual effect of this, right? And like being able to show the breadth there. So I do think we could certainly try to get you some genes. I think the challenge again just is mostly like how much of that data already exists online versus like what is kind of isolated right now. But I think it would be awesome to work on developing more computational methods. I know that, like you mentioned, Rachel Karchin really has been the first one kind of with some of her predictors to try to work on some of that, but it would be great to see some more effort put into the gain of function space for our predictors, just because it is, really challenging to be able to say like, oh, well, can we really trust Revel, for this when it's not really what
I
Revel
don't
was
think it's not
designed for.
designed for that, right?
Yep. And, with one of our SCV-CEPs, we really decided to go a different direction with what Rachel had come up with because it looked like it was going to be just more appropriate.
So besides Rachel's methods, do you know of any other tools that are designed for gain of function in cancer? So there are some gain of function ones also designed for ion channels, but the problem is very different there. I actually don't off the top of my head know of any other specific methods there for gain of function in cancer, and that's just shocking. And there are all these new people coming into the field all the time with their new ideas and their new methods, but I think they look at what's there before and they develop the new thing. And I think where CAGI can really help is again by having this well-posed question that can encourage people to enter the field and try to solve the problem.
Yeah,
Did you
I'm not familiar with other tools. And we have like evaluated quite a few, not hundreds as you were listing, but dozens certainly, with the help of another uh, great tool of Rachel's lab, which is Open Cravat, that does kind of makes it sort of really easy to like get a bunch of predictions all at once for a set of variants. But when you start looking into all those predictors, yeah, they differ in some ways, but mostly they're assessing impact without any like notion of direction. And mostly what you get back is something that seems suitable for loss of function. Um, and that's really like for the ClinGen somatic group, which is kind of closely related to this group. I think we have 8 or 9 expert panels right now. And if you look across what those expert panels have decided to focus on, it's really heavily dominated by oncogenes that predict response, either drive tumors or predict response to particular targeted therapies. Some of them are single gene things like FGFR, and I think it would be really interesting to think about whether there's something specifically that the FGFR group could do that would be synergistic with the FGFR work that you talked about. But just looking more broadly across them, some of them have multiple genes, but even when you look at those multiple genes, those are all oncogenes. And just the pattern of mutations that we're interested in is really um, different from the question that is being asked by Rebel and other predictors like that, which seems like a big problem. And various people have done these analyses where they like do a kind of like a mini version of what you were talking about, like look at every possible amino acid change, and you find this kind of disturbing pattern that some of these oncogenes they tend to be very compact and incredibly conserved, and you just get like really high scores for like 50 to 75% of the amino acids, and it just doesn't jive with your kind of biological expectation that like that something pretty specific is happening for a gain of function mutation. And so it's a really like a needle in a haystack that we're looking for, and the tool doesn't seem to be like giving us good discrimination. The CHASM Plus tool that we have adopted in some of the groups that comes out of Rachel Karchin's lab does seem to kind of focus in on those regions more. But then we've faced the criticism that it's difficult to tease apart how much of that improved prediction is coming from tumor recurrence information, because a few of the features included in that algorithm relate to hotspot information. So sort of empirical observation in other tumors that those sites have been mutated. And I'm not really sure how I think about it. On the one hand, that data is incredibly powerful and useful, so why wouldn't you use it? It makes complete sense. But on the other hand, it's like, I guess there are separate ways to take into account that hotspotness or recurrence information. Um, yeah, so I think that's like a big area of interest for us is could someone develop a tool that was more suitable for our needs for these kind of genes? And we did like recently did a survey, like a landscape survey of the community. Like we have these 8 or 9 expert panels now. Like what would the next like 20 or 30 areas of focus be for clinically relevant curation of variants? And again, they were like still quite heavily dominated by oncogenes. And then fusions are also like a big topic in this area that where like again I'm not aware of any like predictors that like would give you like computational prediction information for a fusion sequence.
Yeah, I agree with that comment, especially I think Steven's something that could be interesting, but I don't know how we'd have to get creative, right? I think it'd be pretty easy for people on this group to come up with a list of genes that have these mutations where there's also like a real dearth of lack of functional evidence, right, supporting them, right? And that was part of what like we submitted to the AVE Alliance was the genes that we were interested in. Just there's not good functional studies for them, right? And so it's like we routinely see these missense mutations pop up and we think they're gain of function, but like nobody's done the tests to like really like prove what we think. And that was part of what, you know, we were interested in really getting out there. So I think we could easily as a community probably put together a list similar to that. It's just how would we then kind of structure like the competition around that and try to get something out of that.
So a few concrete things actually coming from what you just shared. So I actually hadn't realized, I should have known this, but I hadn't realized there's a VCEP that's looking at FGFR. I think it'd be really great to actually connect Russ Altman's group with them, because I think that they would probably have a lot of insights that would help with that assessment and allow that assessment to be more relevant. And so if you can help me make that connection, that would be great. The second thing is you mentioned that you know within the VCEP that they've tried running some different tools. We could actually ask the assessors to actually run some of for FGFR to actually run some of those tools that you found potentially interesting for this purpose you thought might be more relevant for gain of function. That for that list that's been shared with AVE, if we had that list and knew who might be picking those up, if we can know about those before the experiments are even getting going, we could actually probably share those with the predictor community and say these are likely to be coming, and maybe be able to get people thinking about these with a longer lead time. No one can develop a new method for gain of function in the space of a single KG challenge. Our hope is that by saying here's what happened with FGFR, there should be methods that will work for the next KG challenge. But in fact, if we do 2 years in advance that there's going to be some results coming out, that would be an even greater incentive, I think, for people to get involved. And so I think that would be fantastic. You mentioned fusion. As I said, I don't know of any methods for thinking about fusions either. And if there were some data set that might be developed for that type, that would be a great thing to try to incentivize people to develop methods for that. We don't need the 415th method for looking at what a loss of function does. And I think that would have a much greater value for both the field, but also for the people developing the methods to identify and to know where these new areas are. So I think that you're really actually helping the predictors by identifying these overlooked areas as well.
And I do think to that point, Stephen, so the VICC does also have a gene fusion oncogenicity work group right now that's coming up with oncogenicity guidelines for fusion events. As part of our work, I think we could probably provide you with a list of fusion events that would be like good for the competition itself. As we're kind of compiling all of these tricky scenarios when we're getting ready to like test out the existing schema and then work on the publication. I think the group at large would really like to as well put together like a fusion database that these kind of annotations can be added to long-term and kept up to date.
That would be really awesome. And I think something like what we did with Enigma would be really valuable, where, we were able to catch them between, as they were finishing up their classification, but before they made it public, and then have people make predictions. And, it's fine if, if the classification is not done yet, you might have, like 100 possible events you're looking at. And of those, in the end, you might only be able to classify 50 of them. And it's fine if we give people 100 of them, and it turns out they make predictions for 50 of them that can't actually be assessed, if we are able to give them a longer lead time for those 50 that will be assessed. So if there's sort of a hopper of them that are being reviewed, that we could share those out with the community and give them time to work on those. I think for fusion events, that's one also where we'd really want to give the community a long lead time about this, because the methods just don't exist. And we can tell them, hey, we're going to be doing something like this. Here's some examples of things that have been classified in this field before. Here's, maybe even just a couple examples of what type of things we'll be giving you. And we're going to run a challenge, in a year. I think that would be something that might move people into the field. That said, I think that's hard enough that we probably wouldn't move that many people in the field, whereas gain of function, I think is going to be, it's closer to what people are used to doing, but it is so distinct from what's currently underway that I think that we could really help motivate developments.
I will note at this point that Civic has currently 353 fusions curated, about half of which have some evidence for predicting response to a particular therapy, and the majority of those are like an oncogenic relationship, where fusion activates a gene that targets gets inhibited by an inhibitor. So there might be a way to like establish an interesting reference data set from that kind of list. The FGFR group is interesting. They decided ultimately to just go with Revel, and they did that basically by referencing the Pajaver. I think you showed a screenshot of it that was developed for germline purposes, and basically took the stance like it's not for us to like do this kind of algorithm evaluation within an expert panel. We're busy establishing kind of an SOP based on existing methods and recommendations, and we acknowledge that they're not perfect, but this is kind of what we're going to go with for now. I mean, they're really waiting for something like this to happen and then to be published, and then to say this is why you should use CASM Plus or whatever in this context. But I think that group would be very interested in talking about this, and they are currently working through their sort of SOP curation of about 50 specific FGFR3 variants, and maybe those classification results could be an interesting reference point as well.
Yeah, actually, that would be really interesting to do the assessment versus, their classification in addition to against just the experimental data.
In general, like a lot of these groups, so you mentioned like the idea of like predicting in advance and then seeing what gets classified after. I guess some of those classifications will have used predictive algorithms as part of their classification. So there's like potentially like a little bit of a confounding like factor there. But at least with the expert panel curations, you know exactly what was done, as opposed to like a little bit more uncertainty where it's sort of like you have a classification in ClinVar and they called it pathogenic or oncogenic, and it might not be exactly clear to what extent they used computational information to get to that classification and which computational methods they used.
Yeah, I do think that's the potential like fascinating part of the fusions though, right? Because there just isn't any. So like nobody like that's not even part of like our
Yeah.
classification schema right now because it just doesn't exist, right, at all. And so it's
Mhm.
like, can we come up with a schema that we're happy with and then try to come up with in silico predictor that would, correlate with that would be interesting.
Yeah. Looks like
I think
we're at the hour. I think we do have some action items. I think we do want to really connect the FGFR group with you or other folks at CAGI, and the fusion oncogenicity seems like really obvious as well. And then I think we should continue our conversation related to whether there's any data set sort of curation or aggregation in Civic that can be helpful or synergistic as well. And there's probably others as well, but I think this has obviously been a very fruitful conversation. So thank you so much for joining. That was really awesome.
Great. Well, thank you so much. And again, if you have other ideas or connections to make, that would be greatly appreciated. I'll look forward to that connection to the FGR group, learning more about the fusions. And I think also, again, if there were computational methods that you had tried, within, or other groups had tried, we can, ask the assessors for FGFR to just include those as well in their assessment to see, what they do.
Yeah, sounds great.
Thank you.
Great. Thank you very much.